Hi, come on in. Hi, my name is Ryan Miller. I'm a third-year graduate student in the Dr. Kong Research Group. Our lab specializes on the design, characterization, and the application of active biohybrid matter. Our projects primarily focus on studying and addressing vascular degeneration, Alzheimer's disease, disinfection, detoxification of environmental pollutants, as well as the fabrication and assembly of biological robots. Today, I'll be introducing some of our ongoing projects, as well as my lab mates that are working on them. I'll start with my research. I focus on the microfluidic assembly of reactive oxygen species responsive antioxidant crystals. As you can see here, this is a photomask that I use to create a microfluidic chip or a drop maker, as you can see that I have on the microscope stand. From these, I can synthesize various crystal complexes that are used to treat corneal inflammation, as well as detoxify sulfur nanoparticles that accumulate in our aquatic environments. Hi, behind me is Joanne. She's going to be talking about her tough hydrogel project that she's currently working on.
Hi, I'm Joanne. I'm working on tough and stretchable hydrogels. So this is our hydrogel. Unlike conventional ones, ours is really stretchable, which can stretch up to 10 times its original length. And we use it to make organ mimics for surgery practice. We're also looking into its non-drying property and planning to use it in wearable devices and also soft robotics. We can also stretch this hydrogel with our mechanical test machine to measure its actual strength and toughness and other properties. So now my colleague Connie will tell you about her work.
Hi, I'm Connie. So besides the hydrogel research that Joanne just introduced, a big part of our lab work on particle engineering. So my research focused on developing functional particle systems for control release and stem cell engineering. So here's a new batch of particles that I just made using this probe sonicator. So the next step will be a sulfide evaporation for these particles to solidify. So next I will pass on to my colleague Yu-Heng over there. So he's going to talk to you more about his research.
Hello, I'm Yu-Heng. My project is about to fabricate self-propelling particles to clean the biofilm inside PDMS substrate. And here, that's the incubator where I culture the bacteria. So usually I culture bacteria in here and then take out and do some culture things in the biosafety cabinet here. And this is the PDMS substrate cultured with a biofilm, and then I will use self-propelling particles to clean the biofilm inside the PDM substrate. The self-propelling particles I made can convert chemical energy into kinetic energy. For example, in this video, the particles can convert hydrogen peroxide into oxygen bubbles. Those oxygen bubbles can make the particles swim inside the solution. And those particles can penetrate into the biofilm and destroy those biofilm inside the PDMS substrate. So next, my lab mate Caillou will talk about his project.
Hi, my name is Caillou, and I'm doing some in vitro experiment to study the formation of neuromuscular junction. And my project is focused on studying the effect of the engineered substrate on the formation of neuromuscular junction. For example, we will change the topology or the stiffness of the substrate and to see how it affects the interaction between neurons and muscle. So under here is a biohood. So we usually use the biohood to sterilize our stuff, and we can use the UV lamp. And also it has some airflow to prevent like dots or bacteria to go into the hood. So we do the cell culture work inside the hood. Okay, and besides, we have an incubator which we can control the temperature and the percentage of carbon dioxide. So like we usually tune the temperature at 37 degrees Celsius and also 5% of CO2. So that there's an optimal environment for the cells to keep growing. Hi, I'm gonna show you some interesting video. So this is a muscle triggered by electric field. So once this muscle become mature, and we can apply the external electricity field to introduce the contraction of this muscle. So in this video, we are applied a very small electricity, and then we can induce the contraction of muscle. Yeah. And another interesting video is that once the neuromuscular junction is formatted, then we are able to add some chemical like the neurotransmitter to trigger the contractions of the muscle, so that the neurons will interact with the muscles and then start to twitching spontaneously. Then let's head to IGB to use the confocal microscope in the facility to see more details like structure of the cells. Hi everyone, we are now in the IGB facility, and this is LSM700. It's a confocal laser scanning microscope. By using a spatial pinhole, we are able to block the out-of-focus light so that we are able to get an image with higher resolutions and better contrast. And usually you will equipped with several laser beams with different wavelengths. So we can use these laser beams to trigger the fluorescent signals of our fluorescence dye. So here's one example for the neuron stem cells. We culturing these neuron stem cells on our substrate and then differentiate for 7 days. And we use the specific fluorescent dyes with the specific antibody to target neurons and glia cells. So the green color stands for the neuron cells, and the red color stands for the glia cells. And also the red color stands for the nucleus for all cells. So with this microscope, we are able to see what distributions of these cells in our culturing conditions, and also its differentiation level or its differentiation ratio between neurons and glia cells. Thank you for joining this video tour, and hope to see you on campus soon.